So dear students, very good afternoon and welcome back for the today's lecture. I hope that on your screen the title of today's class is already displayed, and even I am sure that I am audible to everyone. So we'll start with today's lecture now. So as you can see on the screen, we'll talk today about Spirogyra sexual mode of reproduction with reference to Scalariform of conjugation. So my friends, all of you are quite aware, in previous few classes we talked much more about asexual mode of reproduction, and in fact in previous class I had told you little bit introductory part about the sexual reproduction in Spirogyra, how it takes place. Now we'll look about Scalariform of conjugation in more detail. So Scalariform of a conjugation, this is very important aspect what we are today going to discuss. You know, the sexual mode of reproduction in Spirogyra is always accomplished by a conjugation. So conjugation, commonly it is called as a fusion mechanism, which is always considered as one of the very primitive method of isogamous type of a sexual reproduction. Now what is isogamous mode of reproduction? You know it very well. Morphological similarity is there, but physiologically they are totally different. This sexual mode of reproduction, especially conjugation, involves the fusion of the entire content of two similar and in fact the unspecialized vegetative cells. When the later cell it becomes yoked. So that is the prerequisite for the sexual mode of reproduction or gamete formation. According to Tiffany, during 1924 he published his research outcome. He suggested that the change of pectose into water soluble pectin increases in the conjugating threads. So please remember, conjugation, this is the one of the important mechanism of a sexual mode of reproduction, and for this purpose there should be a steady situation for a filament of Spirogyra, and it is accomplished by means of a chemical reaction that is called as a conversion of pectose, which is a component of a cell wall of Spirogyra filament, into the soluble pectin condition. So this is the very first investigation which was proposed to create the Spirogyra filament steady. In fact, the Pesoni during 1968 he also recorded that lower concentration of nitrogen and higher light intensity, this also work as one of the important factor to trigger the mechanism of a conjugation. That means at low concentration of nitrogen and higher light intensity it enhances the conjugation process. So sexual conjugation, it takes place toward the end of a growing season. This is one of the important part. So when it takes place toward the growing season's end. There are two strategies by which conjugation in Spirogyra is get accomplished: Scalariform conjugation and lateral conjugation. These are the two basic types. Now as far as concerned with the Scalariform conjugation, it is also known as a ladder type conjugation. It is also referred as a dioecious conjugation because most of the all dioecious species in which filament is actually unisexual, and some monoecious species, very rare cases are there, such as Scalariform conjugations have been get reported. When we talk about the lateral conjugation mechanism, it is also referred as a monoecious conjugation, which happen in case of a bisexual filament, or is simply called as a monoecious species of Spirogyra. So this is very important aspect. You have to keep in your mind these are actually sexual differences. Now when we move ahead for a very first type, that is called as a scalary form of a conjugation, or you simply call it as a ladder form of a conjugation, how it takes place, this part we are going to now discuss. So as you can see on the screen, Scalariform conjugation, alternatively also known as a ladder type of conjugation. It looks like as a ladder shape once it appears toward the entire mechanism. So you have to keep in your mind, it takes place at a night basically, okay, between the recently divided cells. Now how is that particular mechanism going on? This I'll first of all show you in the form of a little bit animated content, and after that we'll discuss in more detail about the Scalariform of a conjugation different phases. So as you can see on the screen, during this Scalariform conjugation, two filaments of Spirogyra species, they come together and lie side by side. The structure which is formed, it looks like as a ladder, and hence this name Scalariform conjugation or H-shaped conjugation has been derived. The tube-like structures, as you can see on the screen, they starts to get developed from each cell of two filaments lying together, and consequently a conjugation canal is formed between these two cells after a fusion of developing conjugation tube. It is followed by the male gamete, its interaction with a female gamete. So here, during a process of a scalariform conjugation, male gamete it fuses with a female gamete of other filament, and one of the filament or filament's cell, it become empty and other has zygotes in its cell. So as you can see on the screen, the consequential stages of scalariform conjugation in schematic way I had tried to keep in front of you. So those zygotes which are produced within that filament, after completion of this mechanism of zygote formation, the zygotes are get released by a mechanism of a cellular decay of parent filaments, and of course the zygote, after undergoing for certain stages of processing, that is generally referred as a zygote maturation and germination process, it produces a new filament under favorable season or favorable conditions. So my friends, I hope that you have understood the essence of a scalariform of a conjugation in this particular step. Just have a look toward the actual stages of this scalariform conjugation, how it is get completed. So single filament is there, it has to be kept in steady position. Now two filaments they come and establishes a conjugation tube, as you can see on the screen. The protoplast of male gamete angium has to be traveled toward the female gamete angium, and process will be followed by zygote formation. So these are the all key events which will be takes place during sexual reproduction, especially scalariform conjugation. Now if we move ahead to look in more detail about scalariform of a conjugation, how it takes place, when it takes place, what are the different stages, these are the points of our discussion. So as you can see on the screen, during night, okay, it takes place. Basically those cells which have recently divided, they are involved in a mechanism of scalariform of a conjugation. And according to the Chapman, the divides, okay, Chapman, according to the Chapman, this particular process is divided into three distinct phases, such as maturation phase, then gammatic union phase, and zygospore contraction phase. So according to the Chapman, these are the three important events taking place while scalariform conjugation is going on. So what are these maturation phase, gammatic union phase, and zygospore contraction phase? We'll see one by one. Let's have a look for a maturation phase. So during maturation phase, the participating filament of the same species of the Spirogyra come together. Morphologically, these filaments are alike. There are no visible differences between them, but functionally they are totally different. So these differences became apparent only at the time of a fusion of a gamete. So morphological differences cannot be seen, but their physiological or functional differences will be found during the gametic fusion process only. So that is the reason why it is called as a isogamous mode of sexual reproduction. Okay, let's move ahead little bit. During the night time, the filaments of these two mating types come closer, come together, and line up side by side and cell by cell, as you can see in the photograph. The single filament does not move at all. So any filament, any one of the filament in this pair, out of that single filament does not move at all. This is very interesting mechanism for scalariform conjugation during maturation phase especially. The presence of other filament, perhaps it act as a stimulus for the first filament. Okay, so the adjusting movements between the conjugation filament, it may either be brought about by a localized secretion of a mucilage or by changes in the surface tension. So if you remember a few minutes back, I have simply told you that the contribution of some phycologists, especially Tiffany, okay, during 1924, he suggested that the change of pectose into water soluble pectin, it seizes in the conjugation thread. So that is the reason why these filaments remains in contact with each other. So nowadays lot of things or factors they have explored out by various phycologists about the maturation phase also, but you have to know about the basic or fundamental things. Now this close contact, okay, is now basically due to a mucilaginous substance or ultimately a surface tension reaction. The close contact, it induces the secretion of more mucilage which hold the filament together, and the sticking together here and there of a pair of filament is the putting out of one important structure that is called as a papilla. So as you can see on the screen now, there will be a formation of papilla. So papilla would be found there properly now. So as you can see now on the screen, there is a rising of a papilla. So papilla are found here. Can you see here? These structures are now referred as papilla. So formation of papilla is taking place now. These papilla, they starts to elongate to form a conjugation processes. Okay, this is a papilla and it goes for elongation. So the conjugation processes of opposite cell, they meet immediately to form the conjugation tube. So as you can see on the screen, a conjugation tube is get established. So the latter elongate and push apart the two conjugating filaments, which are connected all along their length in a ladder like arrangement. So dear students, according to the Lloyd, he states that conjugation papilla of the female cell, okay, at present we cannot say which is female and which is male, but according to the Lloyd, the conjugation papilla of the female cell is usually shorter and thicker as compared to the male papilla. So these are the important findings. There are some species in which male papilla forms the entire conjugation canal also. So these are the some important points on which you have to focus. In fact, I would like to make comments on some important aspects that more often the papilla which are arising from these conjugating filaments, sorry, they are formed on the recently divided cells which are naturally very shorter. There are differences of opinion, of course, with regards to development of papilla. According to the Sonder, a short bird-like papilla arises from one of each pair of opposite cell toward the other. Usually, the papillae appear one of the conjugative filament. On touching these papillae stimulates the development of similar outgrowths from the opposite and corresponding points on the other filament. The papillae are thus in contact from the very beginning. There are some other views in that the papillae simultaneously they grow from the young and very recently formed cells of both the conjugating filaments, as you can see in this diagram B. Similarly, there are some interesting findings which I would like to suggest to you regarding the conjugation canal development mechanism. So the development of papilla according to the result of contact stimulus, generally contact stimulus is called as a thigmotropism. Okay, so papillary development is by means of a thigmotropic mechanism, and thus it is compared to the rhizoids basically. Others hold that they are the result of chemical stimulus also. So Groth during 1977 reported that the Spirogyra, there is one species which is known as a Spirogyra majuscule. Okay, so in this species it grows the conjugating papillae, or is simply called as a copulation tubes over there. It is controlled not by the thigmotropic mechanism, that is touch mechanism or thigmotropic stimuli, but probably by interaction between hormone like chemical substances. So migrating by diffusion from one filament to the other, and depending upon the single division of the cell, these are the controlling factor according to you can call it as a Groth over there. So my dear friends, these are the some interesting findings or detailed investigations related with the papillae formation or conjugation tube formation. Now if you look in more detail, thus based on these all findings, it is said that the freshly agglutinated filament do not yet consist of a gametangia. Here we cannot say which one is a male and which one is a female. we cannot say it is a gametangia. These cells must be termed as a progametangia. So progametangia is the term which is used here for the earliest structure from which gametangium would be get developed. So all progametangial cells, okay, of conjugating filament undergoes for a single division to form a gametangia, and the gametangial cell united by, of course, conjugation tube store abundant amount of a starch. Similarly, the nuclear content, nuclei and nucleoli, diminish in size, and after that there is also a decrease in the permeability and the osmotic pressure of a cell sap starts to happen when progametangia are further process themselves toward the next consequential changes. So these are the some detailed investigations related with the scalariform during scalariform conjugation's maturation phase. What are the reasons or what are the factors uh, responsible to produce or process the conjugation tube over there? Now, with the completion of conjugation tube, the double wall, especially the double wall which is present in between this papillae or conjugation tube, is get dissolved by a enzyme action. Okay, enzymatic action it starts to take place, and of course a continuous passage that is generally called as a conjugation canal, it starts to appear now. So conjugation canal is now get appeared here, and it is of course formed between the two opposite cells as you can see on the screen. The arms of two protoplast, of course protoplast of the gametangial cells which extends into the canal come in direct contact. The two protoplasts remain in contact from this moment onward, and with this particular mechanism a maturation phase in scalariform conjugation is get completed. Let's move ahead for a next step in scalariform conjugation now, and this stage is called as gammatic union phase. Now what is gammatic union phase? During the gammatic union phase, the protoplast of the conjugating cells now called as a gametangia form gametes. Okay, during this phase gametangia are producing gametes. The gametes are formed singly. The protoplast which is destined to functions as a male gamete, it starts to shrink from the parent cell wall without losing a contact with the other female protoplast. So please remember, this contraction is brought about by a removal of water from the contractile vacuole, and of course the rounded male protoplast it migrates through the respective conjugation canal slowly. It joins and fuses with its partner in the opposite cell, which can now be called as a female. So male and female in this way they are get distinguished. The movement of this male gamete is often labeled as the amoeboid movement, but its mechanism however is obscure. Possibly the contact between two protoplasts having been established by dissolution of the intervening walls between the conjugation tubes. Their fusion and rounding may be chiefly due to a surface tension. As you can see in the photograph E, the fusion how it is getting established. It is only after arrival of the male protoplast that the female protoplast contacts, that is the male protoplast that the female contacts from its cell wall only. And of course, the two protoplasts fuse themselves. Their nucleus with nucleus fusion occurs and cytoplasm with cytoplasm. In short, we can call as a karyogamy and plasmogamy takes place with the cytoplasm. And thus, the fusion of two nuclei in certain cases may be get delayed for a while. In certain species it happens, otherwise in most of the cases the nuclear fusion and cytoplasmic fusion almost that takes place at same time. So dear students, this is what one of the important step in a scalariform conjugation, that is called as a gammatic union phase. And of course, during this particular phase, the zygospore contraction phase it arises over there now. So let's move ahead with one important part now, that is zygospore contraction phase. So here zygospore it is get produced. How it is produced you will come to know now. The united protoplast masses, okay, how it enters into the zygospore contraction phase we are going to discuss. Here it is a zygospore produced. So basically zygote of a Spirogyra it undergoes for consequential stages, thereafter it is referred as a zygospore. So let's have a look before going for the I'm sorry, zygospore contraction phase. We'll look for the these major events, how gammatic union phase was get completed under microscopic photographs. You will see here these are the two scalariform conjugating gametangia and their involvement over there. So as you can see here, the migrating protoplast cell is generally called as a male gametangium, or it is called as a active cell, whereas the recipient or receiving one is called as a passive cell or female gametangium. So in this way the moment it takes place and fertilization process is achieved to produce a zygospore. So during scalariform conjugation, the third important stage it is it is arising, it is referred as a zygospore contraction phase now. So zygospore contraction phase, now we will discuss with reference to mechanism of a contractile vacuole actually. So the united protoplasmic masses of two gametes, generally they are called as a zygospore. As you can see, the last phase to be get described is that zygotic contraction, which is more often brought about by a further action of a contractile vacuoles, one of the important cell organelle of the Spirogyra. During this phase of a zygotic or zygospore contraction stage, what happened, the contractile vacuoles of the zygospores protoplast it starts to loses its water, and consequently it starts to shrink away more from the cell wall and it assumes a round or oval form, and a new thick multi-stratose brown wall is get produced around that mass. It is usually differentiated into three layers. Okay, so that particular mass or rounded structure, oval form structure, it differentiates itself into three layers. They are generally called as a exospore, mesospore and endospore. So this is older terminology actually. Basically they are called as a endosporium, then mesosporium and exosporium. So let's have a look for the detail of this exosporium and all the structures. So please look at the extremely outer thin colorless layer of a zygospore wall. It is called as a exosporium, though it is mentioned as a exospore as per the older literature. Now it is called as exosporium. It is made up of a cellulose in nature with pectin predominating chemical constituent. It is smooth but sometimes sculptured, whereas the mesospores, this is the another one, it is the thickest middle layer of the zygospore wall, which shows very interesting mechanism toward the acetylysis and enzyme use that a protesting activity. So acetylysis or enzyme resistance is more often reported or presumably reported due to the presence of a special chemical substance known as a sporopollenin. So sporopollenin, it is present over there, and that's why this actual middle portion of wall is very that a protective in function, and that's why for under any adverse condition this sporopollenin constitution or chemical constitution, it will protect this particular that is zygospore under adverse condition. So it is pale yellow to chestnut brown in color, usually with a dehiscent line. It may be smooth, but sometimes it has sculptures toward the outer surface. So this is the middle portion. Whereas the endosporium, or endospore you can call, is a thin, colorless and innermost layer of a zygospore wall. It is chemically cellulosic in nature. The green zygospore, which is usually ellipsoidal in form, rarely ovoid, or it may be the lenticular in shape. Later on, it becomes darker as the individual chloroplast it starts to get disappear, and the male chloroplasts are usually the first to disappear. The zygospore is now capable of resisting unfavorable conditions, and it appears brownish or red in color, and of course it is densely filled with a starch and oily content. My dear friends, I hope that you have understood this overall mechanism of sexual mode of reproduction in a Spirogyra. So just have a look on the important events under microscope, how it looks. So during scalariform of a conjugation, the mechanism what we have just now seen about the zygospore formation, these are the zygospores, and another filament it totally appears to be like as a empty one, and another the recipient one or female one, it is receiving as it is a recipient one, it looks to be get filled by zygospore. So the in short you can just memorize the cell wall of the original filament, it decay and release the zygospore, and after a resting phase or dormant phase, or after completion of a unfavorable condition, once conditions becomes a little bit in favor to the survival strategy, then these zygospores it germinates and gives rise to a new filament. My friends, now toward the conclusion part of a today's class or lecture, let's let's try to remember some important aspects about gametes of Spirogyra and Zygospore related facts. Let's have a look first of all for the gametes. The entire protoplast of a Spirogyra it act as a gamete. I am talking about this Scalariform conjugation. They are known as a Plano Gametes. They are not distinguished from each other. That's why. A Plano Gametes are formed in the gametangia, which are formed at the end of the growing season of Spirogyra. Whereas Zygospore, it is nothing but the zygote in Spirogyra. The zygospores are basically diploid state of a life during life cycle of this Spirogyra, and it is formed by means of a fusion of a male and female gametes. Zygospores are the only diploid stage in the life cycle of a Spirogyra, and it has a thick wall which is made up of three layers. Just now we have seen they are nothing but the extremely outer one exosporium, middle one is mesosporium, and innermost one is a endosporium. The zygospore it remains dormant until favorable conditions are available, and at the time of a germination these zygospores undergoes a meiosis to form 4 haploid nuclei to maintain the chromosomal or nuclear state of a life. Spirogyra zygospore undergoes a meiotic division, of which only one survive and others they disintegrate, and such a developing zygospore it bursts open to form a germ tube. This germ tube further divides repeatedly by transverse division and develop into a new haploid filament of a Spirogyra. My friends, the further mechanism what happens with this zygospore, how it undergoes a meiotic division, how reduction division it helps to maintain the haploidity of a life cycle in the or the filaments chromosomal status. All these parts we will discuss in next lecture separately in more detail. So this is about one important part about scalariform conjugation. A last part is about scalariform conjugation and differentiation of the sexes. This part also you have to remember. As you know, the Spirogyra gametes which are produced, they do not show any morphological differences, and they are similar in size and form, and therefore they are isogametes which differ only in their behavior. One of them is active, that is called as a male, and moves from one filament to the another, where the second gamete, that is female, awaits for it. The slight distinction of sex between the gametes is accompanied by sexual differentiation between the filaments. The protoplast of all the cells of one conjugating filament act as a male gametes, and those of the other as a female gametes. The filaments are thus unisexual and species referred as dioecious. So the filament that gives off its own protoplast is called as a male, and the one which receives it as a female. Despite this slight distinction of a sex, the reproduction in Spirogyra is considered as isogamous because there are no other visible differences between the gametes and the conjugation filaments. So my friends, this is very important aspect what we have discussed today. Of course, lateral conjugation is the another part. We will discuss it in another video. So thank you very much.